Okay, so thanks again for the second opportunity to get up here and tell you a little about Panaxium. As I mentioned earlier, our team is located mostly in France right now, in Aix-en-Provence. Some of our team is in Toronto, Canada, surrounding Greater Toronto area and expanding. And we have a series of clinical relationships and partnership relationships in Europe, in Canada, and also in the United States. The motivation of our team, so I think a shared one that many of us here have, right, which is that we are focused on some of these big problems in neurocare. For us, we're focused on our eventual objective is therapeutic. And we're focused on, as we said, neuro recovery and things like stroke recovery, traumatic brain injury, and one that has particular interest to us around, in the shorter term, around recovery from damage done through surgical interventions, things like tumor resection procedures. We're very focused, as I just mentioned, we're very focused. This is just kind of the nature of our team and our group and our relationships on tools for the neurosurgeons themselves. So the kind of fundamental, kind of interesting tech that the company has been founded around is this idea of what we describe as an iontronic neural interface. And this is the kind of the exploitation of ion conducting and semiconducting materials. And the idea behind this is that if we microfabricate components using ion conducting materials, you get a much superior, much more kind of natural interface with the brain. And that also gives you some more other interesting features. So one of these is, we're doing this all on kind of very ultra thin polymer substrates. The arrays themselves are very, very thin, just a few microns thin. And we pack in a number of very small sensing devices. We often refer to them internally as electrodes, but they're not really electrodes in a traditional sense. And there's a few factors here. So one, and I'll bravely see what happens if I click on this video. But there's actually a few different things that are going on at once here, which is we actually have the ability. There's three functions here which built into the same device, which is I'll just delete that, make it a little easier. So is that we actually have the ability to pack small molecules into these ion transport materials. And so we can actually create reservoirs of, we describe them as ions, but any kind of polarized molecule, things like neurotransmitters. There's been some demonstration of this using, the collaborators using chemotherapy drugs. That's not our focus, but it's a good demonstrator for different types of molecules and other types of factors for us for kind of neuro recovery. And that's what we've been investigating. But at the same time, in the same kind of pixels, we can also you describe like a conventional electrical stimulation, but functions a little differently. So that using these ion conducting materials, you get a different type of interface. Has a couple advantages. By changing that kind of local ion gradient using a stimulator made out of these materials, you get much lower powers required for the same current injection. And that means we can kind of like boost up the power on these devices, and in doing so, not produce damaging kind of electrochemical byproducts, because you don't get actions as you would on a metal electrode, even at very, very high currents. So all of this gives us kind of interesting advantages we can play with. And then we record to these devices using, in some cases, it looks very much like a conventional electrode, but in some cases with these materials, but we can also make recording devices that are really like little transistors at a pixel. And so all of these combined give you a platform for recording and kind of complex stimulation that is, kind of a mix of really crosses the border between kind of what's electrical and what's chemical when we're working in the brain. And we can place these in hundreds or thousands of them. You can really kind of pack them as small as you want, but it becomes kind of overkill when they're much smaller than kind of like down to the spacing of neurons themselves. It's really not necessary. we can do all of that. And we can do this all up close because we can conform nicely to the sulcine gyre of the brain. And so that's kind of like the kind of foundational technology, but I think we've heard a few comments on this already today that developing the devices is just part of the equation, right? So in order to have a platform for therapeutic procedures, we need to be working on many of these aspects together. And so it became very clear that we need to talk a holistic kind of system-wide approach to therapy. And so while the devices are really an enabler for us, we're developing into our platform aspects of AI-assisted analytics. For that, you kind of need it. And the ability to kind of prepare and collect the data is going to be required to make modulation therapies more confident and easier to deliver as a therapy. And so when we think about what an ultimate kind of neurorecovery therapy will look like in the future, there's a whole bunch of different aspects of this. And some of that comes down to like the device itself and the improved mechanisms of stimulation, but there's other aspects that they're very important. So for starters, in assessment and implantation, this ability to kind of assess the patients, determine what is the best therapy and whether our therapy is appropriate, both in practice, but also throughout the clinical trials and kind of targeting that to demonstrate it the most clarity as possible. It's very special. It's neuromodulation for recovery is very patient specific, and so it's not obvious where to stimulate them when you get up close. And so it's very important for to be able to do that analysis, understand the injury, understand how everything's kind of connected together, and where the best places to kind of plug in the cortex in order to affect it the way you want, and an injury that may be deeper in the white matter. So in Panaxium, or through some of this is through exclusive relationships that we have some partners. We're developing an AI-assisted injury assessment. So this is looking at patients, and this is a proprietary imaging sequences, MRI sequences that are now being collected on stroke patients at 5 centers that our partner is working at now. And another big part of it is that we are jointly developing interoperative tools with a neuronavigation an imaging partner, OEM partner that makes these systems, which will actually be our first commercially released product. That development's underway. We're currently working out the details on building a production facility for clean rooms for our production fabrication of that. And so all of that ability is a key component of this. And then there's a lot of activity when we talk about regenerating new neural cells. There's so much ongoing research around what factors that drive plasticity. We're exploring some of those. I mentioned this in our platform, the ability to deliver transmitters, small molecules, things like stem cell promotion as well. There's a bunch of different factors that we're looking at, and that all can be done on the same devices. And then this is not a new technique, but one that by being able to kind of micro target, idea of remapping connective pathways and doing that with a closed loop system in conjunction with things like physical rehab and the other kind of rehabilitation improvements that are being developed. So this kind of, we see ourselves working on solutions in all of this area to kind of, because we need to, when we look at and see what this looks like as a therapy, we need to be playing in all of this, and we are with either directly or with some very specific partners. I mentioned, in terms of that kind of neurosurgical and neuronavigation systems, we see this as a huge potential for kind of new insights. The idea there to be able to very accurately match up pre-surgical planning and imaging with what we're able to do in the operating suite, and be able to do this high resolution, but yes, large area cortical mapping really is a new area of insights that we can do in real time. And we think that that's going to lead to many new improved insights for future interventions. And we talked about the importance of closed loop feedback. We think that in terms of therapeutic use, that's going to be very important. And this is, like I said, this is a product currently under development. And just as an example, we have several projects underway, but I thought it'd be worth just kind of mentioning one of them. this is one where I think it well represents this idea that where all of the devices are critically enabling to this work, but the points around intelligence and tools in the hands of the clinician is ultimately the objective of this. We're working with Dr. Gilles Luberfeld. This is a study that's been working through, delayed a little bit because of COVID, but starting off, it's kicked off, but the first patients will probably now be very early in the new year, up to 50 glioma and epilepsy patients. And the idea here is to use that type of system that I proposed, along with analysis of high-frequency oscillations, and that in order to give kind of real-time guidance on kind of tumor margin assessment directly in the OR from the electrophysiological recordings. So that's just a bit of a teaser of some of the things we're doing. We just think about, when we're working on this, we have a lot going on. We are looking for investment. We're always looking for investment. There's folks that think that we're an interesting fit. We're still fairly early in our kind of fundraising efforts because we've been able to leverage a lot of government support for our work today. But we're also just looking for partners. I mean, these are big problems. We can't solve them all by ourselves. So I welcome the opportunity for those that have complementary technologies and efforts to seek us out. We're happy and excited to talk, and we look forward to having the opportunity to do so, hopefully here at this event, but anytime afterwards as well. So thank you.
Okay, Brad, if you can stay up at the podium.
Sure.
And moderate from here.
Let me unmute. And can you, yeah, thanks. So I'll open up to questions from the audience in a minute, but I want to delve a little deeper into this ionotronic
Ionotronic is the
ionotronic. Sorry,
yeah.
You're coining a phrase here, so we all got to pronounce it properly. Ionotronic. And by the way, are you the first to use this term?
So we have collaborators I'll give full credit to Linköping University in Sweden, and they have been
okay. So other people are using.
So they've been using it, but
all right. So terminology aside, I want to delve into the technology. So are you able, for instance, to target specific channels within a neural membrane, for instance, a calcium channel versus a sodium channel? And I guess I'm going somewhere with this. I want to see to what extent this might be, a new technology that could be applicable elsewhere besides the brain. So for instance, I'm sure you're aware that Medtronic last year introduced this DTM technology where they're able differentially to attack the cell membrane of a glial cell versus a neuronal cell, which might have completely different membrane characteristics, right? So what's the applicability there?
Yeah, so we talk a lot internally. We've had a lot of conversations, and it's a bit of an academic exercise with our academic collaborators around the ideas of we obviously can do different types of larger molecules, and I should point out is that it doesn't have to be a large molecule. Like we could actually intentionally load up a device with simple ion like potassium or calcium and deliver it in a very kind of like if we wanted to, right? Okay. That's not what we're when we talk about when we can do that. When we talk about this kind of electrical stimulation, we're really doing kind of a non-specific way of doing it, but we've talked about that in terms of both you could do it with different membranes, you can actually do it just by kind of like loading it up with material. And in our case, we're looking at using this for a short-term being still chronic many weeks therapy, but not as a kind of long-term chronic device. So the ability to load it up with even a simple ion that you want to use to deliver, you can imagine doing so. If you do the math, you can actually fit quite a few ions into one of these devices to store them. But we've avoided really the need to be so selective with kind of simple ion channels. So.
Okay, so
Okay, so more specifically, as you progress in your commercialization strategy here, and you're going towards an approval process, would FDA, for instance, see this as similar to a microfluidic product? Would this be a combination product? How would regulators see and regulate this
Yeah, and
type of
we
device?
don't have a clear answer because it's kind of a little bit new territory. In some ways, like the drug delivery, the best kind of examples, I wouldn't say predicates, but are microfluidic delivery systems. So I think that's kind of like the natural thing, and so it becomes a bit of a combination. So in our roadmap, we've actually tried to separate that out, and so that in our kind of therapeutic devices, we have a version of it that is focused first without those ion reservoirs in there, because that's kind of another element, and then we see that as kind of like a secondary approach. So in many ways we're focused on the kind of remapping point, step 3. So step 1 and step 3, and step 2, which is kind of like these endogenous factors and kind of promoting factors for plasticity, are ones that we imagine will be like longer on the roadmap.
Okay, and I have one more question before we open up to other folks. And if you can't answer it, I'll understand. Yeah. But I'm curious whether to date you've had any collaborations with either large medtech firms or large pharmaceutical firms.
So the answer is no. We haven't been. We're open to that, but we've been kind of like just at the point where we feel like in the past 6-9 months that we have all these things kind of worked out, and we've been focused on. We do have this neuronavigation systems partner, and that's been pretty all-consuming for us in terms of our planning. But we'd love to investigate what we can do.
And who's your neuronavigation partner?
I won't publicly disclose that yet.
But it's someone who's already in the neuronavigation space.
Yes.
Okay, that's fine. And in terms of down the road, a year, 2 years, 3 years, would you see your technology more of interest to a large pharma or to a large med tech?
Oh, that's a really great question. I think that I'd love to ask that to the big pharma companies, but I think we see more and more the lines blurring, and that these combination, and when it comes to regenerative medicine as well, like all these kind of like combination therapies as being the way. And the analogy I always say is like, if you're trying to grow a garden, it's not about like if you've got some fertilizer and water and in sunshine, the best, each of those will help your plants grow, but ideally you have all these things working together. And we see is we're kind of like maybe two out of the 3 or 4 things that you want, and the best way to do this is to work with the other components in those combination therapies, and that may be big pharma.
Got it. Okay, sorry, I've been hogging all the questions. Anyone from our online audience who has a question for Brad? Looks like there might be one. No, maybe not. All right, is there anyone from the live audience who has a question for Brad? Yeah, go ahead.
How you release them over time, over the period of a few weeks.
Yeah, and let's repeat that question. I'm not sure the Zoom world heard it.
I think the question is specifically around if you're doing kind of small molecule delivery, how do you protect that from that, and from essentially having it kind of leak out effectively and protect it from the nerves. The answer is that this is an oversimplification. The image here that says these the reservoirs themselves are on the device, but it's not they're controlled because we electrically activate them. We also kind of electrically bias them to keep them from releasing molecules. And we also have some innovations on membrane technologies associated with this, because the initial devices did have some leakage. So like it's minor. It's it's a tiny fraction. But the way that these things were first developed, in academic research, they had a very slow leakage, and depending on the application, that could be an issue. Sometimes it's not, because you want to drive things like with 50,000 times what leaks out. But we've actually worked on improving that kind of protection. Otherwise, there's some membranes that physical barriers that prevent these from getting in.
Okay, any other questions? If not, I'd like to say thank you very much, Brad.
Thank you.